Thank you. Okay, thank you very much, Stephen and the organizers for giving me the opportunity to talk today. For those of you that don't know me, I started at Rutgers New Jersey Medical School a little over 2 years ago. My main focus is structural determination of membrane proteins by cryo-EM. As the title of my talk says, I'm going to be talking today about structural determination of small proteins. This is not necessarily a methods talk, but it's rather that it seems that I like to be working with small proteins. So I will tell you a couple of stories about successful structural determination of small membrane proteins and others by cryo-EM. So I'm going to start with a very brief sort of overview of single particle cryo-EM. I know that most of you are very familiar with the method. Very briefly, the idea is we have our protein sample that is purified through different techniques, and we apply that to a cryo-EM grid. We freeze the protein very fast in liquid ethane to get vitreous size, and then we want to sort of optimize this sample preparation as best as much as possible. So we typically may do some screening runs and back and forth until we get a sample that is up to certain standards. We then go and collect a high resolution data set in a 200 or 300 kilovolt microscope, from which we extract particles, correct them for different aberrations, and then hopefully be able to put them together in good 2D or 2D class averages and 3D reconstructions. So first, I'm going to tell you about essentially one of the main focus of the lab, which is structural determination of enzymes that are responsible in polymyxin resistance. And polymyxins are antibiotics that are used currently to fight multi-drug resistant infections mostly. They are cyclical cationic peptides, and they are thought to work by permeabilizing the bacterial membrane. As most of you will know, gram-negative the gram-negative membrane is consisted of two membranes, inner and outer. The outer membrane is decorated by the bacterial lipopolysaccharide, which is a complex saccharolepid consisted of a lipidic anchor, which is called lipid A, and a complex array of sugars. Lipid A is in fact the point where polymyxin associates with the bacterial membrane, and in order to initiate its action. But of course, bacteria have developed mechanisms to evade this action, and they do so actually by modifying lipid A, essentially capping the two phosphates and modifying the bacterial membrane charge, so the polymyxins, which are cationic, cannot actually associate with the membrane as efficiently. And there are a couple of modifications. We are particularly interested in this modification by aminoarabinose. The reason is that it's also a modification that gives helps bacteria evade more natural cationic peptides. It's an evolutionarily conserved mechanism against antimicrobial peptides, essentially. And we have a complete biosynthetic pathway that operates in gram negatives to essentially synthesize this sugar. So we have the first part of the pathway, which is cytoplasmic, which is the essential synthesis of the sugar. And then the sugar gets loaded on andecaprenyl phosphate, which is the universal sugar carrier in bacteria. And we have another couple of enzymes, a deformylase and a flipase, that act to sort of modify and transport the loaded sugar across the inner membrane, at which point the last enzyme of the pathway, which is Aeron T, picks up the sugar and modifies lipid A to sort of confer the antibiotic resistance, the polymyxin resistance. So I'm going to tell you briefly first about Aeron T, which is sort of spans essentially most of my postdoctoral work as well before joining Rutgers. So my first essentially endeavor in X-ray crystallography and structural determination is the determination of the structure of Aeron T from the extremophile Cryoptus metallidurans. And the way we found this protein was that we used structural genomics principles to sort of look at quite a few different orthologs, and chose one that behaved the best in terms of expression and purification properties. And then we used lipidic cubic phase crystallization to get the crystals in two different states and solve the structure through X-ray crystallography in either the apo state on the left or a partial donor bound state on the right, where we just provided andecaprenyl phosphate without the sugar. So the interesting, there were a few interesting parts. The molecule has a few cavities which seem to be participating in coordination of the two lipidic ligands that it utilizes. And also we observed a very clear conformational transition when the donor substrate binds, where the periplasmic helix shown over here has only 2 turns, but in the bound conformation is extended by an extra 2 turns. And we think that sort of locks the lipidic substrate within the active site. So from this, we only had essentially half of the information, because we knew how the donor potentially binds, but we didn't have much information about the acceptor, which is lipid A. And of course, we observed this big cavity shown over here in purplish color. And it was natural to hypothesize that this is potentially the site that lipid A binds to accept the sugar from the donor, but we didn't know that for sure. So we wanted to find out, and in order to do so, we decided to switch targets, because the identity of lipid A from Cupriavidus is not known, it's not characterized. So we wanted to switch to a species that was more characterized in terms of the lipid A and also more clinically relevant. So we started working with the Salmonella Aeron T. So after some effort, we were able to purify the Salmonella Aeron T, and we tried to crystallize it also. At first, it gave some early crystals, but they diffracted at around 10 angstrom, and we couldn't optimize them any further. So at that point, we decided to go down the cryo-EM route, and we also decided to start working with nanodisk embedding, because that seemed to us like a good approach for delivering lipidic substrates into the enzyme. So I'm sure many of you are familiar, but nanodiscs are essentially discs that are lipid discs that are constrained by scaffold proteins, and you can essentially provide different lipids that are to create the lipid nanodiscs. And in those, you embed your membrane protein. So after some quite a bit of optimization, we were able to obtain first data set of Salmonella Aeron T in nanodisc from a NYSBC Cryos microscope, and this data set behaved extremely well, and we obtained very nice 2D class averages, and it allowed us to create a reconstruction in 3.2 axiom. Mind you, this is a 63 kilodalton molecule, and it's a monomer. So it's essentially, quite close. We consider, the limit of size limitation of cryo-EM, at least a couple of years ago. So I'm going to show you here essentially the reconstruction. We're going to start off with low contours. So you see actually the nanodisc densities. And if we switch to high contour, then you see mostly the protein densities with nice helices, the model overlaid, also with side chains. And we can see many nice hydrophobics. And what came to a surprise to us, and I'm going to show you on the next slide, is this density over here. And this density over here is in fact a ligand density. So although for this first attempt, we didn't add any ligands to the lipidic mix of the nanodisc as we were planning, the protein seems to like to bring the ligands down with it. And because Salmonella Aeron T and E. coli Aeron T are very close in sequence, they seem to be able, they seem to be compatible with the lipids, or cross-species lipids, essentially. So in fact, instead of getting an apostructure, we obtained a doubly bound structure of Aeron T from Salmonella, which was our original target. So we see decapronic phosphate, and we also see a quite clear. Of course, lipid densities are worse than protein densities typically, but we definitely see a very identifiable lipid A density. And moreover, we can essentially, with some confidence, place both the donor phosphate and the acceptor phosphate, and thus we have essentially now defined completely what is our catalytic site, and the distance between the two, although not completely optimal, is within some region. So the other thing that essentially I'm working with right now is to sort of identify what is happening with several conformational changes that I'm seeing. And I'm just going to show you here a comparison between the Salmonella structure, which is the blue one that I just showed you, and the bound metalliduran structure that is essentially just a donor bound structure. So comparing these two structures, we see two very prominent changes, which is one, a complete unwinding of this helix in the periplasmic domain, and also a complete retraction of a loop that was previously sort of projecting within the acceptor binding cavity over here. What we don't know for sure is whether these are in fact changes because of the binding of lipid A, the acceptor, or whether these are changes because we are comparing apples and oranges here a little bit, because we're comparing proteins from two different species. So
time to wrap up, Vasileios.
Yeah.
Thank you.
So I'm going to tell you also about one very recent structure from the lab, which is a different protein in the same pathway, which is Aeron T. And this project we started completely at Rutgers. We were able to purify the protein, and with data that were collected at the Rutgers Arctica, we were able to obtain very nice 2D class averages and a reconstruction at 2.8 angstrom. So I'm going to show you just a little turn video over here. This is a 4 protomer protein, and we were not surprised that this is essentially very similar to a previous glycosyl-transferase GTRB, and the fold is very similar. We just observed a sort of tilt in the transmembrane domain, which we don't know exactly what the significance of that is. But this is a very active project. We just essentially started it. I'm going to skip all this and just show you one essentially other example of a protein we determined in collaboration with my colleague Matt Neiditz at 3 angstrom. This is a soluble transcription factor, and we obtained a peptide-bound structure that was previously not possible by X-ray. This dimer is at 66 kilodalton. And again, these data were obtained at our Arctica microscope in New Brunswick. So the facility is operating, great. So I'm going to wrap it up by thanking everybody in my lab that have worked on many of these parts. I'm going to thank also Matt Neiditz, who has been an excellent collaborator and also is my faculty mentor. My previous postdoctoral mentor, Filippo Mancia at Columbia, NYSBC, Bridget and Clint. Also, of course, Jason Kelber from the Rutgers Cryome facility, Brian Kloss from Compa and structural genomics, cloning and other support. And of course, special thanks to Bill Goes and Vivian Belofato for their support. Thank you very much.